Millions of headless fire ants, scattered around mounds across Texas, Florida, Georgia, 14 states simultaneously. No chemical spill, no visible predator, just bodies and missing heads by the millions. But this isn't a mystery scientists are still trying to solve. They engineered it. And what they unleashed is more precise than anything a laboratory could have designed from scratch. What nobody knew at the time was that a fly smaller than a sesame seed was doing all of this, and that releasing a few thousand of them into open fields would trigger a biological chain reaction still unfolding across the American South today. Stand over a fire ant mound on a summer morning in Texas and look closely enough. The workers are moving differently. Some are running in tight, erratic circles. Some have stopped moving altogether. And scattered at the edges of the mound, there are bodies intact, except for one thing. The head is gone. Not crushed, not torn. Gone as if it was never attached. Multiply that scene across millions of mounds, across 320 million acres of infested land. Scientists don't just suspect what's causing this. They planned it, they bred it, they released it. And the question that still doesn't have a clean answer is exactly how well it's working, and whether something this precise can ever truly be controlled. Solenopsis invicta, the red imported fire ant. It arrived in Mobile, Alabama, sometime in the 1930s, almost certainly as a stowaway and soil ballast dumped from South American cargo ships. Nobody noticed at first. By the time anyone understood what had landed, it was already too late to stop it. Today, fire ants infest more than 14 southern states: Texas, Florida, Georgia, the Carolinas. And they cost the United States somewhere between $6 and $7 billion every year. That figure comes from Texas A and M AgriLife's 2023 update in damage to livestock, crops, electrical infrastructure, and medical treatment for the hundreds of thousands of people stung annually. But the dollar figure undersells the ecological damage. Fire ants displace native ant species that spent millions of years building roles inside their ecosystems. They kill ground-nesting birds. They destabilize soil communities in ways that ripple upward through the food chain. They don't just occupy a niche, they erase the ones already there. And here is the core vulnerability that made everything that follows possible. In South America, fire ants have enemies: parasites, competitors, a whole ecosystem of pressure that keeps their populations in check. In North America, they arrived alone. No natural predators, no parasites that recognize them, just open land and a species with no biological ceiling. For 60 years, that imbalance held. Then, in the late 1990s, researchers decided to close it by importing the one thing fire ants had never encountered on this continent. Something that hunts them from above, lays its eggs inside their bodies, and turns their own workers into the weapon that ends them. The science of classical biological control is, at its core, a detective story. When an invasive species runs rampant in a new land, the first question a smart entomologist asks is not what can we spray on it, and not how do we build a better trap. The first question is, where did it come from? Because somewhere in that original homeland, something is keeping it in check. Some predator, some parasite, some pathogen that co-evolved alongside it over millions of years and learned, through sheer biological pressure, exactly how to exploit it. The pest thrives at home, yes, but within limits. Strip those limits away by moving the pest to a new continent, and you get what happened in the American South: exponential, unchecked explosion. So you go back to the source. By the early 1980s, that is precisely what a USDA Agricultural Research Service entomologist named Dr. Sanford D. Porter began doing. Porter understood that the answer to a North American problem might be buried in the red clay soils of South America, and he was willing to go find it. He and his colleagues mounted field expeditions into Brazil and Argentina, into the native range of Solenopsis invicta, to study what the ant's ecological community looked like when it was whole. What they found was instructive. Fire ants existed there certainly. They were aggressive and numerous and formidable. But they were not dominant in the way they had become in Texas and Florida and Georgia. Something was holding them back. And among the most fascinating of those things were tiny hovering flies, phorid flies, members of the genus Pseudactyon, that Porter and his team began observing with growing excitement. These were not large, dramatic insects. They were barely visible, hovering above foraging columns of fire ants like specks of animated dust, darting in, pulling back, circling again. But their effect on the ants below them was immediate and profound. The flies were parasitoids, meaning they used the ants not merely as a food source, but as a nursery, laying their eggs directly into the ants' bodies and allowing their larvae to develop inside living tissue. This is a relationship that takes millions of years to develop. The fly evolves to exploit a specific host. The host evolves partial defenses. The fly evolves to overcome those defenses. Back and forth, generation by generation, until the two species are locked in a permanent biological negotiation that keeps both populations from spiraling out of control. In South America, fire ants had that negotiation. In North America, they had nothing of the sort. Porter spent years documenting Pseudactyon species, their behaviour, their host specificity, the mechanics of their attack. And what he was building toward, slowly and carefully, was a proposal to import these flies to the United States and release them as a biocontrol agent. The idea was elegant in theory. But the road from elegant theory to actual field release was long, expensive, and fraught with a very reasonable fear. And that wasn't even the part that got people angry. The fear was this: what if the flies, once released, didn't stay where you put them? What if they turned on native ant species, the dozens of non-invasive ants that had evolved in North America and formed the foundation of countless ecological relationships? Classical biocontrol has a dark history. There are cases where introduced predators or parasites jump to unintended hosts and cause damage that dwarfed the original problem. The cane toad in Australia, the mongoose in Hawaii. The USDA was not willing to repeat those mistakes. So before a single fly was released into an American field, years of rigorous quarantine testing were required. Porter and his colleagues exposed Pseudactyon tricuspis, the first species they intended to release, to dozens of native North American ant species under controlled laboratory conditions. They needed to know, with as much certainty as science allows, that these flies would target fire ants and fire ants only. Which, looking back, was either incredibly brave or incredibly stupid, depending on your view of how much certainty science can actually provide about a living organism in a complex open ecosystem. But the testing was thorough, the results were consistent, and by the mid-1990s the evidence was strong enough to move forward. If the researchers had stopped here, this would be a very different story. A careful experiment, a promising result, a file folder of quarantine data sitting in a government archive. But they didn't stop. In 1997, in Florida, Dr. Porter and his team made the first deliberate release of Pseudactyon tricuspis into the wild in the United States. It was not a dramatic moment, at least not visually. A small number of flies released at a site with an established fire ant population allowed to disperse and find hosts on their own. No fanfare, no press conference, just a quiet act with enormous stakes in a warm Florida field, with the sound of insects in the grass and the weight of a decade's work riding on something almost too small to see. The same painstaking caution that built the program's credibility was about to destroy it, or so the critics feared. Because releasing a parasitic fly into the wild is a one-way door. You can study it in a laboratory for 10 years, but the moment it's out there, in the heat and complexity of a living ecosystem, it belongs to the world. and the world does not always behave the way a laboratory predicts. Picture a fire ant mound on a warm October morning in northern Florida, sometime in the early 2000s. The mound is a rough dome of loose soil, maybe 30 centimetres high, sitting in a patch of open ground. Around its base, dozens of foraging workers move in ragged lines, following chemical trails out into the surrounding area. They are small ants, reddish-brown, quick, and they move with the purposeful energy of a colony that is always working. There are hundreds of thousands more of them underground. And above the foragers, so small that a casual observer would never notice, something is hovering. The female Pseudacteon tricuspis is roughly 1 to 2 millimeters long. She is smaller than the head of the fire ant worker she is hunting. She is, by any reasonable measure, almost invisible to the naked eye. A speck, a mote, a thing you might mistake for a trick of the light. But she is not a trick of the light. She is a precision instrument, the product of millions of years of evolutionary refinement, and she knows exactly what she is looking for. She hovers. She watches. The foraging workers below her move in their chemical-guided patterns, unaware, or nearly so, of what is above them. Researchers who have filmed and analysed this behaviour frame by frame note that female phorid flies appear to prefer workers of a certain size, a certain level of activity, a certain positioning away from the main body of the colony. She wants a target that is exposed, isolated enough that surrounding workers won't interfere with what comes next. Then she dives. The entire oviposition event takes less than a second. She drops from her hovering position, makes contact with the foraging worker, and drives a single egg directly through the soft membrane of the ant's thorax using a modified ovipositor that functions essentially as a biological syringe. The ant lurches. The fly is already gone, pulling back into the air before the surrounding workers can mount any effective response. One egg, one ant. One moment of contact so brief it registers almost as an absence rather than an event. But the egg is there now, inside the ant's body. And what happens next unfolds over the following days with a kind of slow, methodical horror that is genuinely difficult to process if you spend too long thinking about it. The egg hatches. The larva that emerges is tiny, soft, and immediately purposeful. It does not stay where it hatched, in the thorax. It begins to migrate, moving through the internal architecture of the ant's body, navigating through tissue and fluid, travelling forward toward the head. This migration takes several days. During that time, the ant continues to function, continues to forage, to follow chemical trails, to perform the tasks the colony requires of it. From the outside, nothing looks wrong. The ant is alive. The ant is working. The colony does not know what is moving through one of its members. By the time the larva reaches the head capsule, it has begun feeding in earnest. It consumes the soft tissues it finds there, muscle, fat body, neural tissue, methodically reducing the interior of the ant's head to raw material. And then it secretes enzymes, specific enzymes, targeted at the membrane connecting the ant's head to its thorax. Those enzymes dissolve that membrane, and the head falls off. Not metaphorically, not approximately. The head of the ant literally detaches from its body and drops to the ground. The ant, such as it is by this point, is no longer an ant in any functional sense. What remains of the body may continue to move briefly. The nervous system doing what nervous systems do in the absence of central coordination, but the creature is finished. The larva, meanwhile, is exactly where it wants to be. The detached head capsule is now a cocoon, a hard, chitinous shell, perfectly sized, offering protection from the environment, from predators, from desiccation. The larva pupates inside it. For 3 to 4 weeks, the empty skull of a fire ant worker serves as the nursery for the next generation of the fly that killed it. And then, when development is complete, an adult fly chews its way out through the ant's face and emerges into the world, ready to begin again. Entomologists who study this system describe it with the careful, measured language of their discipline. But even in that language, the word that comes up most often when describing the larval migration is elegant, which is a generous word for what is, from the ant's perspective, an absolutely catastrophic sequence of events. The clock was already ticking from the moment that first egg was laid in a Florida field in 1997. The fire ants just couldn't hear it yet. But the fly's method, as devastating as it is, does something that no one initially predicted with full clarity. Something that operates not at the level of individual ants, but at the level of the entire colony. Something that changes behaviour, reshapes foraging patterns, and begins to unravel the fire ant's greatest ecological advantage. That effect, the one researchers would come to call the zombie effect, required a different kind of attention to understand, and it would take a few more years and a few more researchers watching fire ant columns in the field before anyone fully grasped what was actually happening. Dr. Lawrence Gilbert saw it coming. He published observations in the early 2000s noting that fire ant colonies in the presence of phorid flies were behaving differently, foraging less aggressively, retreating from food sources, ceding ground to other ant species. Nobody listened, not at first. The implications seemed too large, too strange, too far outside what anyone expected a 2mm fly to accomplish. Picture a Texas pasture in late summer. A red imported fire ant mound rises near a fence post, broad at the base, maybe 18 inches across, built up over months by tens of thousands of workers. On a normal morning, columns of workers stream outward in every direction, following invisible chemical trails to food sources 30, 40, 50 feet away. They move fast, they move with purpose. They are one of the most effective foraging machines in the insect world. Then a phorid fly appears. It doesn't land. It doesn't need to. It simply hovers, a tiny dark speck drifting a few inches above the mound's surface. And within seconds, something remarkable happens. The foraging columns stop. Workers freeze mid-trail or reverse direction entirely, streaming back toward the mound entrance. Some tuck themselves against grass stems. Some disappear underground. Food gets abandoned. Trails go cold. In less than a minute, one of the most aggressive foraging operations in the insect kingdom has simply ceased. The fly didn't sting anyone. It didn't spray anything. It barely moved. It just hovered. And the ants responded as though a silent alarm had gone off across the entire colony. Pay attention to this next detail, because it explains everything. The ants aren't reacting to an attack that has already happened. They're reacting to the possibility of one. Fire ants can detect phorid flies, likely through a combination of visual cues and chemical signals. And the moment that detection occurs, the colony switches modes. Foraging becomes secondary. Self-preservation takes over. And that shift turns out to be one of the most consequential things a 2mm fly has ever caused. Dr. Edward G. LeBrun at the University of Texas at Austin spent years documenting this effect across field sites in Texas and the broader Southeast. What his research confirmed was something the early release teams had suspected but struggled to quantify. The flies weren't just killing ants, they were frightening them, and frightened ants don't eat. A single hovering phorid fly, not a swarm, not a sustained attack, just one fly drifting over a mound, can suppress an entire colony's foraging activity for hours. The workers go underground, go still, go quiet. And while they're doing that, they're not collecting food. Most people never heard about this part. The decapitation story is dramatic enough that it tends to overshadow what comes after. But the behavioural suppression effect is in some ways the more important discovery. Because here's what happens when a colony stops foraging. Nutrition declines, growth slows. The queen, who depends on workers to bring her food, produces fewer eggs. Larvae that need protein don't get enough of it. The colony's reproductive output, its ability to make new queens, new males, new satellite colonies, drops. And it drops not because thousands of workers are being killed. It drops because the workers that are alive are too afraid to do their jobs. Lebrun's field data put numbers to this. Colonies in areas with established phorid fly populations showed significantly reduced foraging activity compared to colonies in areas without them. The effect wasn't marginal. In some cases, foraging suppression was so pronounced that colonies were operating at a fraction of their normal capacity, even when their worker populations looked, from the outside, largely intact. You'd think someone would have said something. Nobody did, at least not loudly enough, not early enough, to shift the public understanding of what the fly program was actually accomplishing. This is the ecological multiplier the researchers kept coming back to. If you release a biological control agent and it kills 20% of a colony's workers, that's meaningful. But if that same agent, through the behavioural changes it triggers, also reduces foraging output by 50 or 60% across the entire season, then the real effect is far larger than the body count suggests. The fly doesn't need to decapitate every ant. It just needs to be present often enough that the ants can never fully relax, never fully commit to the business of growing and expanding and reproducing. And this is where the story takes a turn that even the scientists found striking. Because the behavioural suppression doesn't just hurt the fire ant colony in isolation. It reshapes the ecological landscape around it. Fire ants are dominant partly because they forage so aggressively and continuously that they outcompete native ant species for food and territory. When phorid flies are present and fire ants are suppressing their own foraging, that competitive pressure eases. Native ants, species functionally driven out of areas by fire ant dominance, begin to reappear. They move back into foraging zones the fire ants have vacated. They reclaim food sources. They re-establish themselves in soil ecosystems where they hadn't been seen in years. The fly, in other words, wasn't just a weapon against fire ants. It was, indirectly, a restoration tool for everything the fire ants had displaced. That wasn't the original design. Nobody sat down in a USDA lab and said we're going to use behavioural fear responses to restore native ant diversity across the southeast. It emerged from the biology itself, from a cascade of effects that nobody fully predicted. By the late 1990s, the first release sites were already showing early signs of this shift. Researchers revisiting Texas field sites where Pseudactyon tricuspis had been introduced were noticing changes in anti-community composition. Subtle at first, the kind of thing you'd miss if you weren't counting carefully. But the numbers were there. Native species were moving back in. The fire ants were still present, still numerous. But they were behaving differently, more cautiously, more defensively, less like conquerors and more like a species that had finally, after years of unchecked expansion, encountered something it couldn't simply overwhelm. The fire ant had roughly one breeding season before the next generation of flies would emerge, establish, and begin applying pressure to the next cohort of colonies. And the clock had already started. There is a moment standing at the edge of one of those old Texas release sites when the scale of what was assembled becomes clear. Not the scale of the destruction, that was always obvious, always measurable in crop losses and hospital visits, and the slow disappearance of ground-nesting birds. The scale of the response, 3 species. Sourced from different corners of South America, tested separately, approved separately, released in sequence across a decade of careful, unglamorous fieldwork. Pseudactyon tricuspis came first in 1997. It targets medium-sized fire ant workers, the ones most likely to be out foraging, most likely to be defending the mound's perimeter. It established itself across much of the southeast and became the foundation of everything that followed. Pseudactyon curvatus came later, targeting smaller workers. This mattered because fire ant colonies produce workers across a range of body sizes, each filling different roles. Tricuspis was applying pressure to one part of that workforce. Covatus extended that pressure downward, into the smaller castes the first species largely ignored. Then Pseudactyon obtusus, filling the remaining gaps, ensuring that no worker size, no foraging caste, could move through the world entirely free of parasitism pressure. Each species was sourced from different South American populations and evaluated separately in USDA quarantine facilities before any field release was approved. The researchers weren't just checking whether the flies would attack fire ants. They were checking whether they would attack anything else: native ant species, beneficial insects, organisms that had no part in the problem being solved. The testing was slow. The approvals were slow. The releases were slow. And all of that slowness was, in retrospect, exactly right. Together, the three species create something no single fly could accomplish alone: overlapping pressure across the entire foraging workforce, a layered biological siege that follows the colony through its daily rhythms and seasonal cycles. The flies don't eradicate, they persist, they accumulate, they change the cost of being a fire ant in a way that compounds quietly across years and decades. Back in those early headless fields, the ones that first drew researchers' attention, nobody knew yet that the answer would come in three parts, from three different latitudes, assembled one careful piece at a time. The ants were still winning then. The landscape still belonged to them, but something was already in the air, small, dark, hovering just above the grass. We didn't defeat the fire ant. We found the thing it was already afraid of, and we brought it home. We said we couldn't defeat the fire ant. Turns out, we didn't have to. Subscribe for more comeback stories like this one.